that have been very well probed, so areas like the frontal eye fields, supplementary eye fields, posterior parietal cortex. And those areas, together with parts of the basal ganglion, for example, the caudate nucleus, project down to the superior colliculus, which again, people think of kind of a hub. And I'll talk a little bit about it at the very end of the lecture, but it's a beautiful structure. I think, as you already know, that's really organized as if it was designed for engineers and neurophysiologists to probe. It's got this beautiful mapping where if you stimulate going from the rostral pole to the caudal end, you get bigger and bigger saccades, and if you go off the midline, you increase from pure horizontal saccades on the midline to saccades with either an up component or a down component. So it's just beautiful. And then you can use that electrical mapping to go and record from neurons and see what kind of saccades they're generating. Okay. So I'm going to now move on to really the next stage of processing, and how do you generate the motor command to actually move your eyes. And in particular, eye movements have been very well studied in the, I guess, neuroengineering / neuroscience community, because they have a number of simplifying features which make them pretty, I guess, a nice model system for trying to gain some insight into basic principles. So the first is that there are really, no joints in the system. All you have to deal with the fact is that the eye is rotating, basically, left or right, up or down. You can tort it a little bit. But as opposed to an arm movement where you have multiple joints, it really simplifies the modeling, right? The mechanical modeling of what you need to do to think about how the effector's doing its task, its voluntary task. In addition, as I'll show you as I go through my lectures, the inertia of the eye is relatively negligible, which again simplifies the math and simplifies in turn what the neurons need to do in order to generate accurate control signal in order to produce the eye movement that you want to produce. Okay, so most of what you focused on, I think thus far, has been saccadic eye movements, if I'm correct, right? People have, I guess you've talked about making saccades and error signals for generating accurate saccades. And if you look at the types of eye movements we generate, they're really, the research is basically divides it into 5 subclasses. There are 3 voluntary types of eye movements. Saccades are 1. There are also pursuit eye movements that you can generate to track a moving target. So if you have a visual, imagine watching a tennis match where the ball's not going too fast, you can actually track the ball back and forth. You're using a smooth eye movement to do that. That doesn't have any, has really just smooth, low velocity, continual eye movement. Right? And there's a separate path from the saccadic pathway that generates those eye movements. There are also vergence eye movements. So most of the time, we're not just looking far away and generating conjugate eye movements where both eyes move back and forth like windshield wipers. I'll look at you, then I'll look back at my slides. Every time I do that, one eye is moving a different way than the other eye. I have this vergence eye movement. So the brain also has to control those. And that's something we actually probably know the least about. The work in my own laboratory is try to understand the control of binocular eye movements. But this is an area of continual work. And then we have involuntary eye movements, and I'll note these now because I'm going to talk about the mechanisms that generate them in the next couple lectures. So one is the vestibular ocular reflex, which is one of the essential things that our brain is doing for us all the time without us even noticing it, that keeps us, our sense of space and our movement through space stable in our everyday life. And helping that reflex, because it has a sort of bandpass nature that we'll go over in future lectures, is the optokinetic reflex, which is visual. So you have one reflex that's driven by the vestibular system and another one that's generated by the visual system to keep your gaze stable as you do your everyday activities. So we'll go over those in more detail. Okay. So again, this is just what I mentioned. We have 3 types of voluntary eye movements, saccades, so you can look at them in terms of their eye position, right? So the axes here would be degrees of movement. You move your eye from straight ahead 10 degrees over to the right. That's a 10-degree eye movement. And then there's, of course, the velocity, which would be degrees per second, right, of velocity for a rotational movement. And likewise, you have pursuit. Oops, sorry, this is because I'm working in another format. You also have pursuit eye movements where you have a target that's moving slowly back and forth. So the black line here would be the red dashed line would be the target, and then your eye would follow that target. It steps back a little bit so that you don't get a saccadic component, so that the target's crossing your point of fixation given a visual delay, and visual delays are about 90 milliseconds, the time it takes the target movement to go through all the processing of the retina and all those other stages I showed you to finally make an eye movement. And then of course you have these vergence eye movements where the two eyes move at different amounts. Okay. You can skip this slide. I should have actually pulled it out. This is for another series of lectures where I start with this and then build back out. So what I'm really just going to do is, because you've already heard a bit about how the brain controls eye movements, focus on how the brain's controlling the premotor control of eye movements. There's a reference that you can find online, I guess in Blackboard for you. There's a chapter I wrote that pretty closely follows the first part of or this first lecture in the first part of this chapter. So if you want to go back over and sort of reacquaint yourself with some of the things I've said, this is a very good resource. Okay. So today's lecture is basically broken up into 3 parts. I'm going to first talk about the extraocular muscles and the motor neurons, which I don't think you guys have had any exposure to. Have you? No. Because you might think, well, it's interesting to think about all these high order things like utility functions, et cetera, but how am I actually moving my eyes, right? What kind of motor command is doing it? What kind of constraints does that motor command need to deal with? So first we're going to try to understand just the muscles and the motor neurons that project to them. So it's a little bit of anatomy, really. Some terms. Then understand the motor neuron responses in the context of what we call the extraocular plant, the ocular motor plant, basically the eye that you have to move and all its associated tissues. And then we're going to talk about the implications for control. So that's the way the lecture is organized today. Okay, so if you think about your eye, right, each of your eyes has 6 muscles attached to it. And so there's the front of the eye. You've got 2 muscles that run along, one on the lateral side, one on the medial side. That's the lateral rectus and the medial rectus. And you just imagine pulling on them like a little string, pull one side, pull the other. So if you activate the lateral rectus, you move your eye over towards your ear. If you activate the medial rectus towards your nose. And then complementary to that, you have the superior and the inferior rectus. And they're pretty much the parallel, but for vertical movement. So you activate the superior, the eye moves up, or the inferior and the eye moves down. And then there's these 2 additional muscles that have slightly more complex innervations in the actual eye itself. So you can see this top one, the superior rectus actually goes around this sort of pulley. And what that allows is it torts the eye a little bit too. So you get not just vertical and horizontal movement, but a little bit of torsion. And then there's a complementary muscle down here called the inferior oblique, and you can see it also wraps around such that if you pulled on it, it would tort the eye a bit. Okay. So by activating these 6 muscles to different degrees, you can actually create eye movements, right, and provide the 3-dimensional eye movement behavior that you see in people when they're looking around the world, or monkeys. And again, horizontal, which is the focus of most research because it's really simplified now to just 2 muscles, right? It's unlike arm movements or head movements. To move your head, there are 40 possible muscles involved. For a horizontal eye movement, we can now reduce the problem to 2 muscles. Vertical uses both superior and oblique and regular superior rectus and inferior rectus, so it's 4 muscles. So what I show you in this slide is a cutaway. So you've been focusing on some of the higher parts of the brain, right? Like up in frontal eye fields and posterior parietal cortex, and you'd have deep in here the basal ganglion. But if you go down into the brainstem, right, below where the superior colliculi sit, you have here the superior colliculus on the top of what's called the mesencephalon, the midbrain. And then below that we have these structures which I've given a color code. And this is kind of a useful slide to go back to, just trying to keep track of where everything is. So the 2 red nuclei up here in the mesencephalon are the 3rd nucleus, it's a motor nucleus, and the 4th. They actually have motor neurons in that and that project directly to those muscles I just showed you. So those are nuclei with the neurons that control the muscles that move your eye. And then down here you have the 6th nucleus, which is the abducens. So they're You have a number of cranial nerves. So these are actually the nuclei that have cranial nerves coming out and are hooked up to activate your eye muscles. And in addition, I'll show you a couple other things. I'm going to talk a bit about something called the nucleus prepositus. It's actually back here in the area called VN. VN is for vestibular nuclei. You can think of them as being very close. I'll talk about burst neurons. They sit down here in an area called the PPRF. And then there's basically a little highway connecting neurons in the abducens to neurons up in the third nucleus, and that's called the MLF. So it's a way that you can ensure conjugate eye movements by, say, driving the lateral rectus on this side with the same command as the medial rectus on this side, and that helps your generate your conjugate eye movements. Okay. So in the next slide, I just kind of go through, again, 6 muscles controlled by 3 motor nuclei, abducens. The abducens projects to the lateral rectus, the ocular motor nucleus projects to the medial rectus, and then the trochlear, the abducens projects to the lateral rectus, the trochlear projects to the superior oblique, and the ocular motor projects to all the other muscles. Okay, that's just the way it's organized. So I have down here at the bottom that one of the interesting things is if you look at the motor neurons and you record from them during all those different kinds of eye movements I showed you, they're not dedicated to a particular class of eye movements. They fire during all classes of eye movements. And what I'm going to show you later in the lecture is you can actually take the same model and actually, based on eye movement, predict the responses of a neuron in another context. So there's basically a very fixed relationship between the eye movement, the parameters or kinematics of eye movement, and the way these neurons fire. The only exception to that rule is vergence eye movement. So when you're converging your 2 eyes, there seems to be a failure of general ability to predict. And the thought on that is that you have a ring of smaller neurons around these motor nuclei that people typically do not encounter with their electrodes, and we're undersampling those. And they may be unusually dedicated to slow vergence eye movement. So that would be the one exception. Okay. So again, this is just a little, if I was running my PowerPoint, you'd see it sort of jumping around. But the point, again, sometimes people get a little confused, right, is the saccadic eye movement is the jumping part, and then you fixate between saccades. So you're looking from apple to apple. You look at an apple, that's fixation. You make another jump, that's the actual saccade. And then you can go check the prices out of your apples. Okay, so let's now get to understanding neural responses. I think you've already seen scan paths like this where you can tell someone to look at an image. People will generally look at faces and focus on the eyes. You can give people paintings and ask them to guess how old people are. They'll look at the face, guess them to sort out how wealthy people are. They might look at their shoes and their clothes. So depending on the instructions, people will scan images in a different way. But since these are static images, people will be using saccadic eye movements. So if we want to try to understand how do these motor neurons generate saccadic eye movements, the question is what kind of command do they need to generate in order to get your eye from point A to point B and keep it there, right? That's really what we're trying to understand. And so what we're going to do is a little linear systems analysis, and we've got this very simple system, S, and the input, and it's got an output. And if you were an engineer, you'd want to design this system so that your X input results in what you want as a Y output, right? You're designing that system. But as a neurobiologist, we've been given this system S by, say, evolution. And what we can do is control X and then observe what output we get, right? And that's what and then you can try to guess and see what the system actually is. So that's what we're going to be trying to do. So if you think of the case of the ocular motor plant, right, the question is, what is the output? The output would be an eye movement, right? And the system is the eye muscles, the tissues around the muscles, the orbital tissues, the globe itself, its mass. That's the system. And then the input would be muscle tension, right? What's pulling on the eye. But that's difficult to measure. You can put little strain gauges in, but those actually muck up the eye movements. People have tried that. It's like Joe Miller at UCLA has tried that. But what we can do is actually measure the motor neuron drive to the eye. Okay. And I'm just going to make a note about linearity. I think most people know what linearity is, but one of the sort of constraints in using this type of approach is that the system has to be linear. So you need to keep it in its linear range, and the definition of linearity is simply that if you put two signals in, say this x, and you measure the output y, x1, and then x2, and you measure the output y2, if you put the sum of those two inputs in, you can predict the sum, or predict what the output would be by the sum of the two inputs separately, right? So that's just the simple definition. That's the constraint that is linearity. So we're going to keep the system in a range where it's linear in order to do this analysis. Okay, so if you want to think about how you move your eye, you can think of this sort of basic mechanical type of analysis that people do. And what I'm going to build is a very simple model of a muscle. So what you can see is you can apply a force F, right, to a spring of stiffness K that you think of attached to something. And the stiffer or bigger that string, the more you're going to have to pull it out to a length L, right? That's just basically Hooke's law, right? Which says force equals K, which is a spring constant times the length that you want to pull it out to, right? Then you can think of a second element, right, which would be what's called a viscosity. So you can think of a big hypodermic needle that's filled with oil versus another one that's filled with water, and you're pushing on the piston, pushing the fluid out. And if it's water, you don't need as much sustained force as if it's oil or honey, right? If it was honey, you'd have to push harder and harder, or continue to push harder in order to keep it moving at a constant velocity. So basically that relationship is force equals R, which is the viscosity coefficient, times the velocity at which you want to keep that constant piston motion going. Okay. So what you can do is then take those two elements and put them together, and this is what's called, in parallel, which is called basically a simplified muscle model. And it's called a viscoelasticity, and here you have the force that you're applying shared both by the elasticity and that viscosity. And you end up with a relationship with force being the sum of K times L, right, your spring constant times the length, plus a more dynamic term, which will be your viscosity times the velocity, okay, of your system. So this is effectively, you can see right away, a first order differential equation. So if our system is simple viscoelasticity, we could solve this equation and produce the observed output. So we can get a little more complex, right? And we can now add a mass to this system. And you guys sure remember from Newton's law of motion, right, that force equals ma. So we have a double derivative of length as a function of time, so that's acceleration. So if we wanted to add that into the system, we'd now have something that's a little more complex, which would have basically this elasticity term, the viscosity term, and then this final term, right? And this final term basically now brings us up to a second order differential equation. Okay, so that's just a basic kind of high level review of mechanics, right? Simple mechanics. And so I think you've heard a little bit about Dave Robinson. He was one of the founding fathers of this department, and he did some of the most beautiful work showing the utility of taking basic principles like this and applying them to how the brain actually works and making concrete predictions that he actually verified. And he recently passed away last year. But if you read about the history of our department, he is a very important person, and he really changed the way I think quantitative neuroscience was done. That was very influential. Okay, so again, we can think of this sort of very high-level system and now map it onto our problem of trying to understand how we're going to control the muscles in order to generate accurate eye movement. So what Dave Robinson did is he actually got volunteer students, probably medical students and graduate students, and asked them to come into the lab and do the following experiments. So listen to these, because I don't think we could do these anymore, but they're very pragmatic solutions to sorting out the mechanics, right? If you think about how would I sort out the mechanics of my eye, and what kind of experiment would I do? This seems like a very direct approach. So what he did is he took a section. I don't know if people here wear contact lenses. Probably somebody does. So he put a contact lens, probably thicker than the one you're wearing, on people's eye. It had a little attachment that you could put in a rod that would kind of lock onto a rod so that you could have the subject with this rod in the contact lens on their eye, cover the eye, and have the subject now moving his other eye back and forth to look at different targets. And everything's in depth, so we're not going to worry about Virgin's eye movements. We're assuming all eye movements are conjugate, right? Both eyes are trying to move the same amount. But what he's doing now with this rod connected to the contact lens is holding it fixed, right? So what he effectively can do is measure the torque that's generated as the student tries to move their eye back and forth, because this eye is getting a command to move, but this eye has been blocked by Dave Robinson. So what he can measure here is he's measuring the force, right, as the other eye moves to different amounts. It's kind of cool, right? So this eye is not moving, but he can measure the force it's generating. This eye is moving. So he has a readout of the force that's generated, and he finds in steady state the force equals some constant plus some coefficient times the actual eye rotation. Okay, so that looks a bit like Hooke's law again, right? So then he does another experiment, because he still has this person with a contact lens. Now he's going to tie a little thread onto that attachment area. Instead of take the rod out, tie a thread to it, cover that eye, have the other eye, the seeing eye, stare out onto a fixed target now, and then he pulls on the eye that's not viewing, right, and then lets it go. So if you think about, how fast do you think your eye would go back to center if you took it out and let it go back, right? So what he found is it goes back with a time constant about 200 milliseconds. So it's not that fast. It's not immediate. It has some dynamics, right? It takes it time to get back to center. And what he could do then is fit it with this equation here, and he could compute the C over B, which is effectively the time constant of this equation, and came up with something of about 200 milliseconds. Okay, so we have one last experiment. So in this experiment, again, you've got a contact lens on that subject's eye, and he's going to this time attach a ball and socket joint to it. So this is real engineering, right? And now he's going to attach a little cart on a frictionless sled and put some weight on it so that the eye now has about 100 times the inertia it normally has. And then he asked the subject to make saccades. So you think that this would probably make a really weird looking saccade, but lo and behold, it didn't really alter the saccadic eye movements very much. Okay? So the eye still lands in the right place, and this suggests that the system actually is not really affected by inertia, which makes it a much more simple system to model. And people have gone back and reevaluated this assumption because it's almost too hard to believe, right, that the inertia of the eye wouldn't have a big impact, but it doesn't seem to. So it seems that it holds after all these years. So basically that means we can go back to that simple first order equation. We don't have to go up to a second order equation because the inertia is negligible. Okay, so what we can do is, think about our mechanical systems analysis. We're back to this very simple muscle model that I put up, where the eye plant is really dominated by this viscoelastic properties, where you have the force shared by the elasticity and the viscosity. So that leads to the question of what actually happens when you record from motor neurons. How do they respond during actual movements? Okay, so I'm now going to go through a series of 3 experiments that were done with monkeys a couple years later by Dave Robinson. So these are very simple. So in experiment 1, he just has the monkey look straight ahead. He records from single motor neurons in the 3rd nucleus that I showed you, the ocular motor nucleus. So some are attached to the medial rectus, some to the inferior rectus, right? So there you have the mix of motor neurons. And what he can do is record just the resting discharge, and he finds that the neuron fires at a constant rate. That's very steady state, nice steady state response when the animal's looking straight ahead. And on average, the firing rates are pretty high. They're like 100 spikes per second. Okay, then he does a second experiment. He has the monkey now looking back and forth at targets. And when the monkey looks back and forth at targets, so here we have a neuron that's responding for mostly downward movements, right? It likes when the monkey's eye is down, not up. So you hear the firing action potentials. Neuron turns off when the monkey looks up. So this is probably an inferior oblique muscle, motor neuron, right? And what he can do is, during these points of fixation, right, so here are the saccades where things are changing, but once the eye is settled again in fixation, he measures the eye position, and then he measures the average firing rate, and then say for this neuron, he makes a plot, right? And all those little red dots would be doing that for a bunch of segments, and then he can do that for a bunch of different motor neurons. And look at how the firing rate is correlated with eye position. And what he finds is you get this nice linear relationship that's really quite beautiful. So you can now describe this with firing rate equals some resting discharge, right, which was about 100 from the previous experiment, plus some term which has a coefficient k times the eye position in fixation, right, steady state. Okay, and then he did a third experiment where he actually had the animal now doing that pursuit eye movement I talked about at the very beginning. So he's now making smooth eye movements to pursue something, moving back and forth like this. So you can see an example here, and then you can blow that up. And what he could do is then go into the data and look every time, let's say the eye position was 10 degrees up, measure the firing rate and look at how that varies with eye velocity. So you hold position constant, but you look at places where the eye velocity is different at those same eye positions, and then you can plot that and you get this nice relationship, right? So then we build the second part of this, or the final part of this model, where firing rate is now proportional to some constant times this eye velocity term. So we can put all these together, and based on these three constraints, we end up with this final relationship with firing rate is some resting discharge plus an eye position term plus an eye velocity term. And this provides a really good description of motor neuron activity during all types of eye movements. So you can predict this, say, for saccades, and then use it for pursuit. You can also use it for VOR, vestibular ocular reflex. So it's actually a very robust model. Okay. So again, the good news is that we can go from our mechanical analysis now and come up with a relationship that's also a first order differential equation, right? So we end up with a nice match between Dave Robinson's mechanical analysis in people and the sort of signals that the neurons are actually encoding when he looks at single neurons in alert behaving monkeys. Okay. And the fact that the motor neuron relationship that we just looked at is pretty much the same form as the muscle force equation that he came up with looking at people implies that the relationship between firing rate and muscle force is basically a linear relationship, okay? Because he was measuring muscle force in people and we're measuring firing rate in monkeys, right? So there's a little bit of a gap between those two. That relationship didn't need to be linear. Okay, so what are the implications of that? So if you think about now the kind of signal you're going to want to drive the motor neurons with to produce a saccade, this gives us something to now work with, right? So let's just say you have a step input. So you start at one firing rate and then you move to another firing rate. What would the eye movement look like? So we now have the ability to make that prediction, right? Because we have a nice way to formulate and make predictions about how the neuron should respond for a particular type of input, or how the eye should respond for a particular type of input. So again, this is a first order differential equation. People probably remember from their differential equation course that if you want to solve this, so you flip, instead of looking at firing rate, you put I out on the left side, you end up with a relationship that looks like this, where you basically can then substitute, so T of E would be the time constant, which is effectively R here, this coefficient over the K coefficient. At time, T equals your time constant. This now simplifies to R times 1 minus 1 over E. Then you can continue the math and you end up with R times 1 minus 0.37. So if you want to look and try to understand what this means, what it means is that by, since R and K on average about 1 and 5, you end up with T of E is 200 milliseconds. So what that means is that after about 63, or after about 200 milliseconds, you'll be 63% of your way to your goal, right? So if you think about what a saccade actually looks like, right? Saccades are over in what sort of time? Let's say I make a saccade from here and then 20 degrees over to my right. That saccade is it over faster, or is it faster or slower than 200 milliseconds? How fast are saccades? If I make a 20-degree saccade, anyone know what sort of time frame where saccades occur in? It'd be more like 50 milliseconds max. You'd be on target, right? So you're generating saccadic eye movements super fast. So this would be, if you just had a step input, you would be kind of creeping your way over to that endpoint. It would not be very good, right? You'd be generating these kind of slow, sluggish movements. Okay. So it would be too slow. Saccades, even big saccades are over within 100 milliseconds. The example I gave you is probably 20 milliseconds. So what happens? So what Dave Robinson predicted is that you had to have some sort of pulse, initial kick, to get the eye moving, and then you had this step to then keep it at its new position. So the idea is that you then have to have two types of commands, right? And that's what's shown here. The pulse, this would be the proposed or predicted input. So here's the pulse and here's the step. And if you think about it, the pulse looks a lot like the velocity of a saccade, and the step looks a lot like the position of a saccade. So you can kind of simplify it to the pulse being kind of a velocity type signal and the pulse being more of a position signal. Okay, so again, we need this extra burst to drive the saccade and complete a saccade in a shorter time to overcome the viscous drag. And the idea is that's produced by a particular group of neurons. And what we have actually been able to do as neurophysiologists is go into the brain, and remember that area I showed you in blue on one of the very first slides called the PPRF? That has neurons that only burst during saccades. They're off during fixation, they burst during the saccade, and then they're off again when you're at the new target. So they really have what looks like a velocity signal. And then you also need this step, and the question is, how are you going to get a step, right? How is the brain going to make this step? So if you think about it from a math point of view, we know we have a signal in the PPRF that looks like velocity, right? So what he proposed is that there's a structure that then takes that input and integrates it, right? Which sounds like kind of an engineer's dream, and you can draw that on a napkin. But indeed, that seems to be really what is the case. So again, we know that the pulse is produced by these burst neurons, and people had recorded from them. So the hypothesis is there's a neural network that then integrates that pulse and turns it into the step that we need. Okay, so here's what I was referring back to. So there's this area called the PPRF, and that's where the burst neurons sit. So, and these are the burst neurons. The system's organized in a really beautiful way. If you go into this structure, those burst neurons encode just the horizontal component of the saccade, the horizontal velocity. And if you go up here into these areas in green, they're taking care of torsion and vertical. Okay, so the system is kind of almost surprisingly clean in the way it's divvied things up to match onto the way you've got your eye muscles sorted out. So the burst, the PPRF neurons are purely horizontal and deal with the medial and lateral rectus, whereas the neurons up in the MRF, up in the midbrain, are dealing with the other four muscles. Okay, and then what was discovered was behind the abducens, right? Remember, that's one of the horizontal motor neuron pool, those are the neurons that project to the lateral rectus, is a structure called the nucleus prepositus. And if you actually put an electrode into those neurons, you find neurons that actually encode, they have a bit of a burst, but they also encode eye position fixation. So you have basically projections from the PPRF over to the nucleus prepositus, and the nucleus prepositus has a network within it that helps to integrate the signal. Okay. And you can show that both by recording, but then to really show that it's causal, right, that it's creating this tonic signal you need to keep your eye in the place where you want to keep it when you're fixating that price over the apples, right? What they did is they looked at a monkey and had him generate normal saccades, right? So this would be normal horizontal and normal vertical saccades. And then they made an injection of, I forget, it was muscimol, probably muscimol, something that would inhibit the neurons so that they didn't fire, and looked and see. So it's a temporary lesion, effectively. so that the neurons are not activated anymore. And what you can see is the monkey can now make saccades, right? He can look left and right, but the problem is once he's looking over to the left or right, his eye drifts back because he no longer has this component of the drive that's going to basically compute this step. Right? He's got the pulse because we didn't hit the PPRF neurons, but he doesn't have the step anymore. Okay. And the question is, how do you do this? Or how does the brain do it? And again, this is the pulse that looks like velocity. So if you record from these PPRF neurons, they just encode a burst during saccades that's graded with velocity. More spikes, the faster you're moving your eye. They project directly to both the motor neurons and to the nucleus prepositus. And then if you go within the nucleus prepositus, there are networks basically so that the neurons are reciprocally connected. And what that does is cause a form of memory, right? So if you have a burst of input going in here, it activates this neuron, but then it goes around this loop. And that gives it, right, staying power. So this is basically a little model of working memory. If people look at, try to model how do you remember a phone number for a little bit, you'll come up with basically these models where you have reciprocal loops, right? It keeps things active, and depending on how you set the gain, you want the gain lower than 1 or it's going to blow up. Since it's going to be a little lower than 1, it's going to wind down over time, right? Okay. And then here I put this back together with something you already know, I think, from previous lectures, right? So how does this now link back to areas like the superior colliculus? So you have, this is the little circuit I just showed you. Burst neurons project both to the motor neurons and to the nucleus prepositus. Nucleus prepositus has this recurrent feedback in it that's basically a form of memory that holds that burst and turns it into a position signal. So it's a form of integration. And it also projects to the motor neurons, and we know that. And then basically the way you drive the burst neurons is from those, that map I mentioned on the superior colliculus, right? So you have a weighted progression from the caudal projections are stronger than the rostral projections. So you get a big burst if the back part of the colliculus is activated, where you represent very big saccades, and a smaller burst if you activate the more rostral part. And then there's this very cool additional thing that the very rostral pole is sort of the inverted logic of the burst neurons. So the burst neurons burst when you make a saccade. You have this sort of check system called omnipause neurons. Have you guys heard about those at all? No? So like the burst neurons, they burst during the saccade. The omnipause neurons shut off. And then once you're on fixation, the burst neurons are off and the omnipause neurons are on. So you can think of the pause neurons as being active fixation neurons. They tell you when you're fixating. And if you look at the most rostral part of the superior colliculus, it similarly has neurons that are called fixation cells. So people typically think of the saccades as being an active movement, but fixation itself is active. It's just basically organized by a slightly different network. And you basically have reciprocal inhibition so that you're either, the question is, should I stay and keep looking at something, or should I go? And you have a balance of power from the cortex that's going to basically have sort of dueling activity in the rostral versus the caudal colliculus to determine if you're going to keep looking at something, if you're going to look at something else. Okay. And I'm just going to finish by noting for the nucleus prepositus, it may all sound like some sort of engineer's fantasy, but people have now actually gone in using different types of approaches. So David Tank's lab at Princeton's done really beautiful work in the zebrafish, where they've gone in and used techniques like calcium imaging to watch a network of cells do the integration as, you might not think much about zebrafish eye movements, but they make pretty respectable eye movements, and you can actually watch activity as this circuit is integrating. And my own group, we can now put in these laminar probes that allow us to record high density from multiple neurons within the network, and we can look and see basically the connectivity of the network by looking at near neurons versus far neurons, and try to understand the connectivity as a function of distance away from the input, and try to understand how that integration is actually occurring in real time by looking across groups of neurons. So again, both by using calcium imaging and now by using these higher density probes that are becoming more and more standard, and I guess more and more sophisticated for use in both non-human primates and rodents. So I think I'll stop there, and I think we're really at time. So I'm happy to take any questions, and I'll put up the lectures for the vestibular system lecture on, I guess, Friday in the next little bit. Okay, thank you.